DNA is the blueprint of biological life from its inception to its growth until the end. The discovery of its structure explained how it could replicate and how it stores and transmits heredity. This video is going to focus on DNA replication. DNA is made up of two chains that wrap around each other in the shape of a double helix. The backbone of DNA is based on a repeated pattern of a sugar and a phosphate group. DNA has four complicated organic bases that form nucleotides. Those nucleotides are adenine, thymine, guanine, and cytosine. Adenine is a purine base that is constituent involved in base pairing with thymine in DNA. Thymine is a pyrimidine base that is an essential constituent of DNA. Guanine is a purine base that is an essential constituent of both RNA and DNA. Cytosine is a pyrimidine base occurring in animal and plant cells. It usually is condensed with ribose or deoxyribose to form nucleotides. There are three steps to DNA replication. Helicase separates the DNA strands, almost like a zipper. Then that forms the replication fork. DNA polymers bind to a single strand of DNA. This adds complementary nucleotides and forms a new strand. DNA ligase bonds phosphate to the deoxyribose. This forms the DNA backbone. DNA polymers makes mistakes at a rate of about 1 per every 100,000 nucleotides. In humans, with our 6 billion base pairs in each diploid cell, that would amount to about 120,000 mistakes every time a cell divides. Some of the mistakes are corrected immediately during replication through a process known as proofreading, and some are corrected after replication in a process called mismatch repair. Proofreading fixes about 99% of these types of errors, but that's still not good enough for normal cell functioning. Then it is the turn of the mismatch repair. During mismatch repair, enzymes recognize and fix these deformities by removing the incorrectly paired nucleotide and replacing it with the correct nucleotide. Mutation is a change in the nucleotide sequence of a DNA molecule. For example, instead of pairing up with cytosine, guanine pairs up with thymine. When these mistakes are not corrected, the incorrectly sequenced DNA strand serves as a template for future replication events, causing all the base pairings thereafter to be wrong. These may cause a cancer.